right, so we're going to start part 2 of our cell theory and organelle notes. This is on page 5 of your note packet. And we're going to start with the Golgi apparatus. So it is a folded membrane. So think of like a crepe folded up on itself or layers of pancakes stacked together. And its job is to get vesicles of protein from the ER. Now, vesicles are like little vehicles. So they're like mini carts and they transport proteins around the cell. So they get proteins from the endoplasmic reticulum and they take them to the Golgi apparatus. And then the Golgi apparatus is kind of like your shipping center. So it processes, sorts, and then sends the proteins on wherever they are needed. And these are found in our eukaryotic cells or our animal and plant cells, but not bacteria. Then we have lysosomes. This is a really exciting picture. It's a ball. And it contains enzymes. We're going to talk a lot about enzymes later on, but its entire job of a lysosome is to break down dead stuff. So our lysosome just likes to eat dead things. And that means that it will break down dead food, bacteria, and old parts of the cell. And it can also be programmed to do apoptosis. Apoptosis. Let me get back to you on that one, which is essentially programmed cell death. So if something is going wrong within the cell, then the lysosome can kind of take over and say, we are not good for our job anymore, and can kind of just say, all right, bye. And the cell is done. And these are only found in our animal cells. So we have these, but plants do not. Then our vacuoles. So we have little vacuoles in animal cells. So like those little spots up there by the word cell. And then we also have one large central one in plants. So you'll see the plant cell has it looks like a big swimming pool. You could think of the vacuole like a swimming pool, but essentially its job is storage. So it just holds in water and nutrients and waste, and it is not found in our bacteria cells. So our centrioles and centrosomes looks just like a sun and some long sticks wrapped together. They're made of microtubules, which is essentially, try to think of just a bunch of sticks, really, that are all kind of condensed together, and they will sometimes form a little ball shape, and they show up during cell division. So their entire job is to help divide cells by pulling our chromosomes apart, and we'll get to that a lot more in mitosis at the end of this unit. But the centrioles and centrosomes are just waiting for the cell to divide. And they are in our animal cells, but not our plant or our bacteria cells. Cilia and flagella. So these they're fun. They're just little tiny things, and I think a lot of students forget about them. But cilia, it's like little tiny hairs, kind of think about a peach with fuzz on it. And they actually, all those little fuzz pieces act like little oars. So that's how our cell moves around. Now, flagella are longer and there's a lot less of them. So a flagella is kind of just like a fishtail. And the cilia moves fluid across the cell surface. So it's got little tiny oars all over the peach, and it's just moving the water around and away. But flagella, because it's just one and so powerful, like a fishtail, it moves the entire cell around. So that's how our bacteria is always moving. And you'll see it on the bacteria picture. That's your flagella. And cilia is not usually pictured because it is tiny little peach fuzz all over our cell in our animal cells only. Mitochondria, the mighty mitochondria. It has two different parts. So you have the inner membrane, which is that squiggly part, and then the matrix, which is the green fluid part that you see around it. Is it actually green? We'll find out. And its job, this is where cellular respiration happens. This is how your cell breathes to break down food and releases energy in the form of ATP. We will get into this a lot later on. But cellular respiration cannot happen without mitochondria. And we get our energy from our food. So, like if you eat your carbs because you're trying to carb load before a race, we don't just use carbs directly. We have to break them down, and inside the carbs, we have ATP, and that is what our body uses to actually energize us. And it's considered the powerhouse of the cell because it provides power. And our mitochondria is in our eukaryotic cells. Chloroplast. It's got two parts. I like to call them pancakes and syrup. So your grana are the stacks of green in the middle right there. And then your stroma is the syrup or the fluid that is around it. And this is where photosynthesis happens, which is essentially the plant version of cellular respiration. So it takes energy from the sun and turns it into energy in the form of sugar. So it only happens in our plant cells. If we could get energy from the sun, that would be super cool. And your cell wall is made of cellulose, which is essentially fiber in plants, and chitin, I think, in fungi. We're not really going to worry about that one too much. I would prefer that you just remember cellulose is in plants, and then peptidoglycan in bacteria. And its entire job is to protect and maintain the shape. So we have our cell wall around our plant cells and our bacteria, but our animal cells have the cell membrane only. Then our central vacuole, we already talked about vacuoles, but this is just one giant one right in the middle, and it's just a big storage center. So this is like a giant warehouse where everything is for the cell, and it is that big thing in the middle of our plant cell. So we did already talk about that when we talked about vacuoles, but here it is again, just as a friendly little reminder. So now you have on page 7 and 8, I want you to fill these out to submit to Google Classroom and then turn into me by next Monday or Tuesday, whenever I see you. And you are going to be filling out this Venn diagram. So if you're not sure how to fill out a Venn diagram, let me walk you through that real fast. Get my camera up. Okay, so in our Venn diagrams, what we're doing here is we have our prokaryote on this side, eukaryote on this side, and everything that they share is in the middle. So whatever we can tell that our prokaryote and eukaryote cells have in common, we're going to put in this middle. If we only find it in a prokaryote cell, we're going to put it over on the left. And if we only, find it in eukaryote, we're going to put it on the right. Same thing with our animal cells and plant cells. There are organelles that they share. So we're going to put the ones that they both have in the middle, and the ones that only are in animal cells on the left, and the ones that are only in plant cells on the right. Then you are going to complete this practice organelle identification, and using all the different organelles we have talked about, you're going to write their names in on this 1 through 15. So page 7 and 8, you will need to have done for Google Classroom and class.